The chance observation made by Pasteur and his collaborator that culture of Pasteur aviseptica, which has been left in the laboratory during lost its virulence for chicken, and chicken injected with such aged culture were protected against re-challenge with virulent strain. This process of rendering virulent microbe avirulent by aging or other means is called attenuation. The protection given to animal by pre-inoculation with such attenuated microorganism lead to the widespread use of this method of immunization purposes. The idea of immunization using attenuated strains was reconfirmed by Pasteur experiment on anthrax in 1891. To the honor of the work of Jenner nearly 100 years before, Pasteur extended the term vaccination to denote the state of immunity that developed following injection of attenuated microorganisms. Until that time, the biological changes in the animal body and the basic principle underlying this acquired protection were unknown. But a year later, in 1882, Mechnikov, a Russian zoologist, attributed immunity state to the phagocytosis cellular theory of immunity. Immunology as a science began with the demonstration made by von Behring and Kitsato in 1890 of the presence of antibacterial substance of factor in the blood of animal immunized against diphtheria and tetanus toxoid. The neutralizing antibody of such blood factor for bacterial toxin was the first demonstration to what is known as antibody humoral theory of immunity. Almort Wright reconciled the two opposing theory, cellular versus humoral, by the phagocytosis process. He showed that coating of the microbes with antibody facilitated the engulfing and digestion of the microorganism. This process was called opsonization, and the antibody in this case was considered as opsonins. Progress in the field of immunology continued. Pfeiffer and Bourdet discovered a substance in the serum of the animal, immunized or not, with which complete the effect of antibody molecule and has lytic effect on the antigen coated with specific antibody. This substance is called the complement. Long time after the above mentioned discoveries, in 1960, and during investigation on allergy, a completely new form of immunity was identified. It's the cell-mediated immunity that depend upon lymphocyte cell population. It's now clear that the word immunity refers to a state of resistance to infection by particular micro